Good afternoon, students. Today we will have a lecture in renal physiology, and we will be discussing such subtopics today. So first, anatomic functional features of the urinary system, functions of the kidneys, microanatomy of the kidneys, the structure of the nephron vascular system of the kidneys, glomerular filtration and its mechanisms, control of glomerular filtration rate, the method of clearance and its diagnostic value, tubular reabsorption and its mechanisms, countercurrent system of the kidneys and its role in the concentration of urine, tubular secretion and its role in the homeostasis of the body, and urine analysis and its interpretation. So let's start about functions of the kidneys. So the kidneys perform many specific functions, most of which help preserve the constancy of the internal fluid environment. So first function of the kidney is to maintain water balance in the body. Next, maintaining the proper osmolarity of body fluids, primarily through regulating water balance. This function is important to prevent osmotic fluxes into or out of the cells, which could lead to detrimental swelling or shrinking of the cells, respectively. Third function, regulating the quantity and concentration of most extracellular fluid ions, including sodium, chloride, potassium, calcium, hydrogen ion, bicarbonate, and phosphate. Also sulfate and magnesium, etc. Even minor fluctuations in the extracellular fluid concentrations of some of these electrolytes can have profound influences. For example, changes in the extracellular fluid concentration of potassium can potentially lead to fatal cardiac dysfunction. Next function, maintaining proper plasma volume, which is important in the long-term regulation of arterial blood pressure. This function is accomplished through the kidney's regulatory role in salt and water balance, helping maintain the proper acid-base balance of the body by adjusting urinary output of hydrogen ions and bicarbonate, excreting or eliminating the end products of bodily metabolism, such as urea from proteins, uric acid from nucleic acids, creatinine from muscle creatine, bilirubin from hemoglobin, and hormone metabolites. If allowed to accumulate, many of these wastes are toxic, especially to the brain. Next one, excreting many foreign compounds such as drugs, food additives, pesticides, and other exogenous non-nutritive materials that have entered the body. Producing erythropoietin, a hormone that stimulates red blood cell production. Producing renin, an enzymatic hormone that triggers a chain reaction important in salt conservation by the kidneys. And the last one is converting vitamin D into its active form. So the urinary system consists of the urine-forming organs, the kidneys, and the structures that carry the urine from the kidneys to the outside for elimination from the body. The kidneys are a pair of bean-shaped organs that lie behind the abdominal cavity, between the abdominal cavity and the back muscles. One on each side of the vertebral column, slightly above the waistline. Each kidney is supplied by the renal artery, a renal vein, which respectively enters and leaves the kidney. At the medial indentation that gives this organ its bean-like form. The kidney acts on the plasma flowing through it to produce urine, conserving materials to be retained in the body and eliminating unwanted materials into the urine. After urine is formed, it drains into a central collecting cavity, the renal pelvis, located at the medial inner core of each kidney. From there, urine is channeled into the ureter, a smooth muscle-walled duct that exits at the medial border close to the renal artery and vein. There are two ureters, one carrying urine from each kidney to the single urinary bladder. The urinary bladder, which temporarily stores urine, is a hollow, distensible, smooth muscle-walled sac. Periodically, urine is emptied into the bladder to the outside through another tube called the urethra as a result of bladder contraction. The urethra in females is straight and short, passing directly from the neck of the bladder to the outside. In males, the urethra is longer and follows a curving course from the bladder to the outside, passing through both the prostate gland and the penis. The male urethra serves the dual function of providing both a route for eliminating urine from the bladder and passageway for semen from the reproductive organs. The prostate gland lies below the neck of the bladder and completely encircles the urethra. The functional unit of the kidney is nephron. Each kidney consists of about 1 million microscopic functional units, as we said previously, nephron. Nephrons are bound together by connective tissue. Recall that a functional unit is the smallest unit within the organ capable of performing all of that organ's functions. Because the main function of the kidneys is to produce urine and in so doing maintain constancy in the extracellular fluid composition, a nephron is the smallest unit capable of forming urine. The arrangement of nephrons within the kidneys gives rise to two distinct regions. An outer region called the renal cortex, which looks granular, and the inner region, the renal medulla, which is made up of striated triangles, the renal pyramids. Each nephron consists of a vascular component and tubular component, both of which are intimately related structurally and functionally. Let's talk about vascular component of the nephron. So the dominant part of the nephron's vascular component is the glomerulus, a ball-like tuft of capillaries through which part of the water and solutes is filtered from blood passing through. This filtered fluid, which is almost identical in composition to plasma, then passes through the nephron's tubular component, where various transport processes convert it into urine. On entering the kidney, the renal artery subdivides and ultimately form many small vessels known as afferent arterioles, one of which supplies each nephron. The afferent arteriole delivers blood to the glomerulus. The glomerular capillaries rejoin to form another arteriole, the efferent arteriole, through which blood that was not filtered into the tubular component leaves the glomerulus. The efferent arterioles are the only arterioles in the body that drain from capillaries. Typically, arterioles break up into capillaries that rejoin to form venules. At the glomerular capillaries, no oxygen or nutrients are extracted from the blood for use by the kidney, nor are waste products picked up from the surrounding tissue. Thus, arterial blood enters the glomerular capillaries through the afferent arteriole, and arterial blood leaves the glomerulus through the afferent arterioles. The efferent arteriole quickly subdivides into a second set of capillaries, the peritubular capillaries, which supply the renal tissue with blood and are important in exchanges between the tubular system and the blood during conversion of the filtered fluid into urine. These peritubular capillaries, as the name implies, are intertwined around the tubular system. So the peritubular capillaries rejoin to form venules that ultimately drain into the renal vein by which blood leaves the kidneys. Tubular component of the nephron. The nephron's tubular component is a hollow, fluid-filled tube formed by a single layer of epithelial cells. Even though the tubule is continuous from its beginning near the glomerulus to its ending at the renal pelvis, it is arbitrarily divided into various segments based on differences in structure and function along its length. The tubular component begins with Bowman's capsule, an expanded double-walled cup that surrounds the glomerulus to collect fluid filtered from the glomerular capillaries. The presence of all glomeruli and associated Bowman's capsules in the cortex produces this region's granular appearance. From Bowman's capsule, the filtered fluid passes into the proximal tubule, which lies entirely within the cortex and is highly coiled or convoluted throughout much of its course. The next segment, the loop of Henle, forms a sharp U-shaped or hairpin loop that dips into the renal medulla. The descending limb of the loop of Henle plunges from the cortex into the medulla. The ascending limb traverses back into the cortex. The ascending limb returns to the glomerular region of its own nephron, where it passes through the fork formed by the afferent and efferent arterioles. Both the tubular and the vascular cells at this point are specialized to form the juxtaglomerular apparatus, a structure that lies next to glomerulus. This specialized region plays an important role in regulating kidney function. Beyond the juxtaglomerular apparatus, the tubule again coils tightly to form the distal tubule, which also lies entirely within the cortex. The distal tubule empties into a collecting duct or tubule, with each collecting duct draining fluid from up to 8 separate nephrons. Each collecting duct plunges down through the medulla to empty its fluid contents, which now converted into urine, into the renal pelvis. The parallel arrangement of long loops of Henle and collecting ducts in the medulla creates this region's striated appearance. So the three basic renal processes are glomerular filtration, tubular reabsorption, and tubular secretion. Now let's talk about glomerular filtration. So fluid filtered from the glomerulus into the Bowman's capsule must pass through the 3 layers that make up the glomerular membrane. So first, the glomerular capillary wall consists of a single layer of flattened endothelial cells. It is perforated by many large pores or fenestrations that make it more than 100 times more permeable to water and solutes than capillaries elsewhere in the body. And the second is the basement membrane, which is an acellular, which means lacking cells, gelatinous layer sandwiched between the glomerulus and the Bowman's capsule. The inner layer of Bowman's capsules, the third component, consists of podocytes, octopus-like cells that encircle the glomerular tuft. Each podocyte bears many elongated food processes that interdigitate with food processes of adjacent podocytes, much as you interlace your fingers between each other when you cup your hands around the ball. The narrow slits between adjacent food processes, known as filtration slits, provide a pathway through which fluid leaving the glomerular capillaries that enter the lumen of Bowman's capsule. Thus, the route that filtered substances take across the glomerular membrane is completely extracellular, first through capillary pores, then through the acellular basement membrane, and finally through capsular filtration slits. Collectively, these layers function as a fine molecular sieve that retains the blood cells and plasma proteins, but permits water and solutes to filter through. Let's talk now about the glomerular capillary blood pressure, which is the major force that causes glomerular filtration. So to accomplish glomerular filtration, a force must drive a part of the plasma in the glomerulus through the openings in the glomerular membrane. No active transport mechanisms or local energy expenditures are involved in moving fluid from the plasma across the glomerular membrane into Bowman's capsule. Passive physical forces similar to those acting across capillaries elsewhere accomplish glomerular filtration. Because the glomerulus is a tuft of capillaries, the same principles of fluid dynamics apply here that cause ultrafiltration across other capillaries, except for two important differences. First, the glomerular capillaries are more permeable than capillaries elsewhere, so more fluid is filtered for a given filtration pressure. Second, the balance of forces across the glomerular membrane is such that filtration occurs the entire length of the capillaries. In contrast, the balance of forces in other capillaries shifts so that filtration occurs in the beginning part of the vessel, but reabsorption occurs toward the vessel's end. Three physical forces are involved in glomerular filtration. These are glomerular capillary blood pressure, plasma colloid osmotic pressure, and Bowman's capsule hydrostatic pressure. Let's examine the role of each. So first, glomerular capillary blood pressure. This is the fluid or hydrostatic pressure exerted by the blood within the glomerular capillaries. It ultimately depends on contraction of the heart and the resistance to blood flow offered by the afferent and efferent arterioles. Glomerular capillary blood pressure at an estimated average value of 55 millimeters mercury is higher than capillary blood pressure elsewhere. The reason for the higher pressure is the larger diameter of the afferent arteriole compared to that of the efferent arteriole. Because blood can flow more rapidly into the glomerulus through the wide afferent arteriole than it can leave through the narrower efferent arteriole, glomerular capillary blood pressure is maintained high as a result of blood damming up in the glomerular capillaries. Also, because of the high resistance offered by the efferent arterioles, blood pressure does not have the same tendency to fall along the length of the glomerular capillaries as it does along the other capillaries. This elevated non-decremental glomerular blood pressure tends to push fluid out of the glomerulus into Bowman's capsule along the glomerular capillary's entire length. And it is the major force producing glomerular filtration. Whereas glomerular capillary blood pressure favors filtration, the two other forces acting across the glomerular membrane oppose filtration. So second is plasma colloid osmotic pressure. This pressure is caused by the unequal distribution of plasma proteins across the glomerular membrane. Because plasma proteins cannot be filtered, they are in the glomerular capillaries, but not in Bowman's capsule. Accordingly, the concentration of water is higher in Bowman's capsule than in the glomerular capillaries. The resulting tendency for H2O to move by osmosis down its own concentration gradient from Bowman's capsule into the glomerulus opposes glomerular filtration. This opposing osmotic force averages 30 millimeters mercury, which is slightly higher than across other capillaries. It is higher because more H2O is filtered out of the glomerular blood, so the concentration of plasma proteins is higher than elsewhere. And the third force is Bowman's capsule hydrostatic pressure. The pressure exerted by the fluid in this initial part of the tubule is estimated to be 15 millimeters mercury. This pressure, which tends to push fluid out of Bowman's capsule, opposes the filtration of fluid from the glomerulus into Bowman's capsule. The total force favoring filtration is the glomerular capillary blood pressure at 55 millimeters mercury. So the total of the two forces opposing filtration is 45 millimeters mercury. So the net differences favoring filtration, 10 millimeters mercury of pressure, is called the net filtration pressure. This modest pressure forces large volumes of fluid from the blood through the highly permeable glomerular membrane. Normally, about 20% of the plasma that enters the glomerulus is filtered at the net filtration pressure of 10 millimeters mercury, producing collectively through all glomeruli 180 liters of glomerular filtrate each day, for an average glomerular filtration rate of 125 millimeters per minute in males and 115 millimeters per minute in females. Changes in the glomerular filtration rate result primarily from changes in the glomerular capillary blood pressure. Because the net filtration pressure that accomplishes glomerular filtration is simply the result of an imbalance of opposing physical forces between the glomerular capillary plasma and Bowman's capsule fluid. Alterations in any of these physical forces can affect the glomerular filtration rate. We now examine the effect that changes in each of these physical forces have on the glomerular filtration rate. So first, unregulated influences on the GFR. Plasma colloid osmotic pressure and Bowman's capsule hydrostatic pressure are not subject to regulation and under normal conditions do not vary much. However, they can change pathologically and those inadvertently affect the glomerular filtration rate. Because plasma colloid osmotic pressure opposes filtration, a decrease in plasma protein concentration by reducing this pressure leads to an increase in the glomerular filtration rate. An uncontrollable reduction in plasma protein concentration may occur, for example, in severely burned patients who lose a large quantity of protein-rich plasma-derived fluid through the exposed burnt surface of the skin. Conversely, when plasma colloid osmotic pressure is elevated, such as in cases of dehydrating diarrhea, the GFR is reduced. Bowman's capsule hydrostatic pressure can become uncontrollably elevated, and filtration subsequently can decrease given a urinary tract obstruction, such as kidney stone or enlarged prostate. The damming up of fluid behind the obstruction elevates capsular hydrostatic pressure. Sympathetic control of the GFR is aimed at long-term regulation of arterial blood pressure. If plasma volume is decreased, for example by hemorrhage, the resulting fall in arterial blood pressure is detected by the arterial carotid sinus and aortic arch baroreceptors, which initiate neural reflexes to raise blood pressure toward normal. These reflex responses are coordinated by the cardiovascular control center in the brainstem and are mediated primarily through increased sympathetic activity to the heart and blood vessels. Although the resulting increase in both cardiac output and total peripheral resistance helps raise blood pressure toward normal, in the short term, plasma volume is still reduced. In the long term, plasma volume must be restored to normal. One compensation for a depleted plasma volume is reduced urine output so that more fluid than normal is conserved for the body. Urine output is reduced in part by reducing the GFR. If less fluid is filtered, less is available to excrete. Tubular reabsorption. This is a highly selective process. All constituents except plasma proteins are at the same concentration in the glomerular filtrate as in plasma. In most cases, the quantity reabsorbed of each substance is the amount required to maintain the proper composition and volume of the internal fluid environment. In general, the tubules have a high reabsorptive capacity for substances needed by the body and little or no reabsorptive capacity for substances of no value. Accordingly, only a small percentage, if any, of filtered plasma constituents that are useful to the body are present in the urine, most having been reabsorbed and returned to the blood. Only excess amounts of essential materials, such as electrolytes, are excreted in the urine. For the essential plasma constituents regulated by the kidneys, absorptive capacity may vary depending on the body's needs. In contrast, a large percentage of filtered waste products are present in the urine. These waste, which are useless or even potentially harmful to the body, if allowed to accumulate, are not reabsorbed to any extent. Instead, they stay in the tubules to be eliminated in the urine. As H2O and other valuable constituents are reabsorbed, the waste products remaining in the tubular fluid become highly concentrated. Of the 125 millimeters of fluid filtered per minute, typically 124 millimeters per minute are reabsorbed. Considering the magnitude of glomerular filtration, the extent of tubular reabsorption is tremendous. The tubules typically reabsorb 99% of the filtered water, 100% of the filtered sugar, and 99.5% of the filtered salt. Tubular reabsorption involves transepithelial transport. Throughout its length, the tubule wall is 1 cell thick and is close to a surrounding peritubular capillary. Adjacent tubular cells do not come into contact with each other except where they are joined by tight junctions at the lateral edges near the luminal membranes, which face the tubular lumen. The interstitial fluid lies in the gaps between adjacent cells, the lateral spaces, as well as between the tubules and the capillaries. The basolateral membrane faces the interstitial fluid at the base and lateral edges of the cell. The tight junctions largely prevent substances from moving between the cells, so materials must pass through the cells to leave the tubular lumen and gain entry to the blood. To be reabsorbed, a substance must go across 5 distinct barriers. First, it must leave the tubular fluid by crossing the luminal membrane of the tubular cell. Second, it must pass through the cytosol from one side of the tubular cell to the other. Third, it must cross the basolateral membrane of the tubular cell to enter the interstitial fluid. Fourth, it must diffuse through the interstitial fluid. And the last one, it must penetrate the capillary wall to enter the blood plasma. This entire sequence of steps is known as transepithelial transport. The two types of tubular reabsorption, passive and active, depends on whether local energy expenditure is needed for reabsorbing a particular substance. In passive reabsorption, all steps in the transepithelial transport of a substance from the tubular lumen to the plasma are passive. That is, no energy is spent for the substance's net movement, which occurs down electrochemical or osmotic gradients. In contrast, active reabsorption takes place if any step in the transepithelial transport of a substance requires energy, even if the 4 other steps are passive. With active reabsorption, net movement of the substance from the tubular lumen to the plasma occurs against an electrochemical gradient. Substances that are actively reabsorbed are of particular importance to the body, such as glucose, amino acids, and other organic nutrients, as well as sodium and other electrolytes, such as phosphate ions. Rather than specifically describing the reabsorptive process for each of the many filtered substances returned to the plasma, I will give examples of the general mechanisms involved after first highlighting the unique and important case of sodium reabsorption. So an active sodium-potassium ATPase pump in the basolateral membrane is essential for sodium reabsorption. Sodium reabsorption is unique and complex. Of the total energy spent by kidneys, 80% is used for sodium transport, indicating the importance of this process. Unlike most filtered solutes, sodium is reabsorbed throughout most of the tubule, but this occurs to varying extents in different regions. Of the sodium filtered, 99.5% is normally reabsorbed. Of the sodium reabsorbed, on average, 67% is reabsorbed in the proximal tubule, 25% in the loop of Henle, and 8% in the distal and collecting ducts, tubules. Sodium reabsorption plays different important roles in each of these segments. So sodium reabsorption in the proximal tubule plays a pivotal role in reabsorbing glucose, amino acids, water, chloride, and urea. Sodium reabsorption is in the ascending limb of the loop of Henle, along with chloride reabsorption, plays a critical role in the kidney's ability to produce urine of varying concentrations and volumes, depending on the body's need to conserve or eliminate H2O. Sodium reabsorption in the distal and collecting tubules is variable and subject to hormonal control. It plays a key role in regulating extracellular fluid volume, which is important in long-term control of arterial blood pressure and is also linked in part to potassium secretion. Sodium is reabsorbed throughout the tubule with the exception of the descending limb of the loop of Henle. Throughout all sodium reabsorbing segments of the tubule, the active step in sodium reabsorption involves the energy-dependent sodium-potassium ATPases carrier located in the tubular cell's basolateral membrane. This carrier is the same sodium-potassium pump present in all cells that actively extrude sodium from the cell. As this basolateral pump transports sodium out of the tubular cell into the lateral space, it keeps the intracellular sodium concentration low while it simultaneously builds up the concentration of sodium in the lateral space. That is, it moves sodium against a concentration gradient. Because the intracellular sodium concentration is kept low by basolateral pump activity, a concentration gradient is established that favors the passive movement of sodium from its higher concentration in the tubular lumen across the luminal border into the tubular cell. The nature of the luminal sodium channels and transport carriers that permit movement of sodium from the lumen into the cell varies for different parts of the tubule, but in each case, movement of sodium across the luminal membrane is always a passive step. For example, in the proximal tubule, sodium crosses the luminal border by a symport carrier that simultaneously moves sodium and an organic nutrient such as glucose from the lumen into the cell. By contrast, in the collecting duct, sodium crosses the luminal border through a sodium leak channel. Once sodium enters the cell across the luminal border, by whatever means, it is actively extruded to the lateral space by the basolateral sodium-potassium pump. This step is the same throughout the tubule. Sodium continues to diffuse down a concentration gradient from its high concentration in the lateral space into the surrounding interstitial fluid and finally into the peritubular capillary blood. Thus, net transport of sodium from the tubular lumen into the blood occurs at the expense of energy. First, let us consider the importance of regulating sodium reabsorption in the distal portion of the nephron and examine how this control is accomplished. So active sodium reabsorption is responsible for the passive reabsorption of chloride, H2O, and urea. Not only is secondary active reabsorption of glucose and amino acids linked to the basolateral sodium-potassium pump, but passive reabsorption of chloride, H2O, and urea also depends on this active sodium reabsorption mechanisms. So negatively charged chloride ions are passively reabsorbed down the electrochemical gradient created by the active reabsorption of the positively charged sodium ions. For the most part, chloride ions pass between, not through, the tubular cells through leaky tight junctions. The amount of chloride reabsorbed is determined by the rate of active sodium reabsorption instead of being directly controlled by the kidney. Water is passively reabsorbed throughout the length of the tubule as H2O osmotically follows sodium that is actively reabsorbed. Of the water filtered, 65%, that is 117 liters per day, is passively reabsorbed by the end of the proximal tubule. Another 15% of the filtered H2O is obligatorily reabsorbed from the loop of Henle. This 80% of the filtered water is reabsorbed in the proximal tubule and Henle's loop, regardless of the H2O load in the body, and is not subject to regulation. Variable amounts of the remaining 20% are reabsorbed in the distal portion of the tubule. The extent of reabsorption in the distal and collecting tubules is under direct hormonal control, depending on the body state of hydration. No part of the tubule directly requires energy for this tremendous reabsorption of water. During reabsorption, water passes primarily through aquaporins, or water channels, formed by specific plasma membrane proteins in the tubular cells. Different types of water channels are present in various parts of the nephron. The water channels in the proximal tubule are always open, accounting for the high H2O permeability of this region. The channels in the distal parts of the nephron, in contrast, are regulated by the hormone Vasopressin, accounting for the variable water reabsorption in this region. The medullary vertical osmotic gradient is established by countercurrent multiplication. We now follow the filtrate through a long-looped nephron to see how this structure establishes a vertical osmotic gradient in the medulla. Immediately after the filtrate is formed, uncontrolled osmotic reabsorption of filtered water occurs in the proximal tubule secondary to active sodium reabsorption. As a result, by the end of the proximal tubule, about 65% of the filtrate has been reabsorbed, but the 35% remaining in the tubular lumen still has the same osmolarity as the body fluids. Therefore, the fluid entering the loop of Henle is still isotonic. An additional 15% of the filtered water is obligatorily reabsorbed from the loop of Henle during the establishment and maintenance of the vertical osmotic gradient, with the osmolarity of the tubular fluid being altered in the process. The following functional distinctions between the descending limb of a long Henle's loop, which carries fluid from the proximal tubule down into the depth of the medulla, and the ascending limb, which carries fluid up and out of the medulla into the distal tubule, are crucial for establishing the incremental osmotic gradient in the medullary interstitial fluid. So the descending limb is highly permeable to water via abundant, always open water channels. It does not actively extrude sodium, that is, it does not reabsorb sodium. It is the only segment of the entire tubule that does not do so. The ascending limb actively transports sodium chloride out of the tubular lumen into the surrounding interstitial fluid and is always impermeable to water, so salt leaves the tubular fluid without H2O osmotically following along. So now let's talk in detail about mechanism of countercurrent multiplication. The proximity and the countercurrent flow of the two limbs allow important interactions between them. Even though the flow of fluids is continuous through the loop of Henle, we can visualize what happens step by step. So initially, before the vertical osmotic gradient is established, the medullary interstitial fluid concentrations is uniformly 300 milliosmol. Now please look at the top left picture. So as are the rest of the body fluids, 300. The active salt pump in ascending limb can transport sodium chloride out of the lumen until the surrounding interstitial fluid is 200 milliosmol more concentrated than the tubular fluid in this limb. When the ascending limb pump starts actively extruding sodium chloride, the medullary interstitial fluid becomes hypertonic. Water cannot follow osmotically from the ascending limb because this limb is impermeable to water. However, net diffusion of water does occur from the descending limb into the interstitial fluid. The tubular fluid entering the descending limb from the proximal tubule is isotonic. Because the descending limb is highly permeable to water, net diffusion to water occurs by osmosis out of the descending limb into the more concentrated interstitial fluid. The passive movement of water out of the descending limb continues until the osmolarities of the fluid in the descending limb and the interstitial fluid become equilibrated. Thus, the tubular fluid entering the loop of Henle immediately starts to become more concentrated as it loses water. At equilibrium, the osmolarity of the ascending limb fluid is 200 milliosmol, and the osmolarities of the interstitial fluid and descending limb fluid are equal at 400 milliosmol. You can see this at step 1 of this slide. If we now advance the entire column of fluid in the loop of Henle several frames, look at the step 2 at the slide, a mass of 200 milliosmol fluid exits from the top of the ascending limb into the distal tubule, and a new mass of isotonic fluid at 300 milliosmol enters the top of the descending limb from the proximal tube. At the bottom of the loop, a comparable mass of 400 milliosmol fluid from the descending limb moves forward around, around the tip into the ascending limb, placing it opposite a 400 milliosmol region in the descending limb. Note that the 200 milliosmol concentration difference has been lost at both the top and the bottom of the loop. The ascending limb pump again transports sodium chloride out, while water passively leaves the descending limb until a 200 milliosmol difference is re-established between the ascending limb and both the interstitial fluid and the descending limb at each horizontal level, as it's pictured at the step 3. Note, however, that the concentration of tubular fluid is progressively increasing in the descending limb and progressively decreasing in the ascending limb. As the tubular fluid is advanced still further, step 4, the 200 milliosmol concentration gradient is disrupted again at all horizontal levels. Again, active extrusion of sodium chloride from the ascending limb, coupled with the net diffusion of H2O out of the descending limb, reestablishes the 200 milliosmol gradient at each horizontal level. Step 5. As the fluid flows slightly forward again, and this stepwise process continues, you can observe this at step 6. The fluid in the descending limb becomes progressively more hypertonic until it reaches a maximum concentration of 1,200 milliosmol at the bottom of the loop, 4 times the normal concentration of body fluids. Because the interstitial fluid always achieves equilibrium with the descending limb, an incremental vertical concentration gradient ranging from 300 to 1,200 milliosmol is likewise established in the medullary interstitial fluid. In contrast, the concentration of the tubule fluid progressively decreases in the ascending limb as sodium chloride is pumped out, but water is unable to follow. In fact, the tubular fluid even becomes hypotonic before leaving the ascending limb to enter the distal tubule at a concentration of 100 milliosmol, 1/3 the normal concentration of body fluids. Note that although a gradient of only 200 milliosmol exists between the ascending limb and the surrounding fluids at each medullary horizontal level, a larger vertical gradient exists from the top to the bottom of the medulla. Even though the ascending limb pump can generate a gradient of only 200 milliosmol, this effect is multiplied into a large vertical gradient because of the countercurrent flow within the loop. This concentrating mechanism accomplished by the loop of Henle is known as a countercurrent multiplication or countercurrent mechanism. We have artificially described countercurrent multiplication in a stop and flow stepwise fashion to facilitate understanding. It is important to realize that once the incremental medullary gradient is established, it stays constant because of the continuous flow of fluid, coupled with the ongoing ascending limb active transport and the accompanying descending limb passive fluxes. Now let's move on and discuss the next process, which is tubular secretion. Like tubular reabsorption, tubular secretion involves transepithelial transport, but now the steps are reversed. By providing a second route of entry into the tubules for selected substances, tubular secretion, the discrete transfer of substances from the peritubular capillaries into the tubular lumen, is a supplemental mechanism that hastens elimination of these compounds from the body. Anything that gains entry to the tubular fluid, whether by glomerular filtration or tubular secretion, and fails to be reabsorbed is eliminated in the urine. The most important substances secreted by the tubules are hydrogen ion, potassium ion, and organic anions and cations, many of which are compounds foreign to the body. Hydrogen ion secretion is very important in acid-base balance. Renal hydrogen secretion is extremely important in regulating acid-base balance in the body. Hydrogen ion secreted into the tubular fluid is eliminated from the body in the urine. Hydrogen ion can be secreted by the proximal, distal, and collecting tubules, with the extent of hydrogen secretion depending on the acidity of the body fluids. When the body fluids are too acidic, hydrogen secretion increases. Conversely, hydrogen secretion is reduced when the hydrogen concentration in the body fluids is too low. Potassium ion secretion is controlled by aldosterone. So potassium is one of the most abundant cations in the body, but about 98% of the potassium is in the intracellular fluid, because the sodium-potassium pump actively transports potassium into the cells. Because only a relatively small amount of potassium is in the extracellular fluid, even slight changes in the extracellular fluid potassium load can have a pronounced effect on the plasma potassium concentration. Changes in the plasma potassium concentration have a marked influence on membrane excitability. So let's talk about mechanism of potassium secretion. Potassium ion secretion in the distal and collecting tubules is coupled to sodium reabsorption by the energy-dependent basolateral sodium-potassium pump. This pump not only moves sodium out of the cell into the lateral space, but also transports potassium from the lateral space into the tubular cells. The resulting high intracellular potassium concentration favors net movement of sodium from the cells into the tubular lumen. Movement across the luminal membrane occurs passively through the large number of potassium leak channels in this barrier in the distal and collecting tubules. By keeping the interstitial fluid concentration of potassium low, as it transports potassium into the tubular cells from the surrounding interstitial fluid, the basolateral pump encourages passive movement of potassium out of the peritubular capillary plasma into the interstitial fluid. A potassium ion leaving the plasma in this manner is later pumped into the cells, from which it passively moves into the lumen. In this way, the basolateral pump actively induces the net secretion of potassium from the peritubular capillary plasma into the tubular lumen in the distal parts of the nephron. Because potassium secretion is linked with sodium reabsorption by the sodium-potassium pump, why isn't potassium secreted throughout the sodium reabsorbing segments of the tubule instead of taking place only in the distal parts of the nephron? The answer lies in the location of the passive sodium leak channels. In the distal and collecting tubules, the sodium channels are concentrated in the luminal membrane, providing a route for potassium pumped into the cell to exit into the lumen, those being secreted. In the other tubular segments, the potassium leak channels are located primarily in the basolateral membrane. As a result, potassium pumped into the cell from the lateral space by the sodium pump sodium-potassium pump simply moves back out into the lateral space through these channels. This potassium recycling permits the ongoing operation of the sodium-potassium pump to accomplish sodium reabsorption with no local net effect on potassium. Now let's talk about plasma clearance. So of the 125 milliliters of plasma filtered per minute, typically 124 milliliters per minute are reabsorbed. So the final quantity of urine formed averages 1 milliliter per minute. Those of the 180 liters filtered per day, 1.5 liters of urine are excreted. Urine contains high concentration of various waste products, plus variable amounts of these substances regulated by the kidneys, with any excess quantities having spilled into the urine. Useful substances are conserved by reabsorption, so they do not appear in the urine. A relatively small changes in the quantity of filtrate reabsorbed can bring about a large change in the volume of urine formed. For example, a reduction of less than 1% in the total reabsorption from 124 to 123 milliliters per minute increases the urinary excretion rate by 100% from 1 to 2 milliliters per minute. So plasma clearance is the volume of plasma cleared of a particular substance per minute. Compared to plasma entering the kidneys through the renal arteries, plasma leaving the kidneys through the renal veins lacks the materials that were left behind to be eliminated in the urine. By excreting substances in the urine, the kidneys clean or clear the plasma flowing through them of these substances. The plasma clearance of any substance is defined as the volume of plasma completely cleared of that substance by the kidneys per minute. It refers not to the amount of the substance removed, but to the volume of plasma from which that amount was removed. Plasma clearance is actually a more useful measure than urine excretion. It is more important to know what effect urine excretion has on removing materials from body fluids than to know the volume and composition of discarded urine. Plasma clearance expresses the kidney's effectiveness in removing various substances from the internal fluid environment. The plasma clearance rate varies for different substances depending on how the kidneys handle each substance. So if the substance is filtered but not reabsorbed or secreted, its plasma clearance rate equals the glomerular filtration rate. Assume that a plasma constituent, for example, substance X, is freely filtered at the glomerulus, but is not reabsorbed or secreted. As 125 milliliters per minute of plasma are filtered and subsequently reabsorbed, the quantity of substance X originally contained within the 125 milliliter is left behind in the tubules to be excreted. Those 125 milliliters of plasma are cleared of substance X each minute. Of the 125 milliliters per minute of plasma filtered, 124 milliliters per minute of the filtered fluid are returned through reabsorption to the plasma minus substance X, thus clearing this 124 milliliters of substance X. In addition, the 1 milliliter per minute of fluid lost in urine is eventually replaced by an equivalent volume of ingested water that is already clear of substance X. Therefore, 125 milliliters of plasma cleared of substance X are, in effect, returned to the plasma for every 125 milliliters of plasma filtered per minute. No normally occurring chemical in the body has the characteristics of substance X. All substances naturally present in the plasma, even wastes, are reabsorbed or secreted to some extent. However, inulin, please do not confuse with insulin, these are two different substances. So inulin is a harmless foreign carbohydrate. It's freely filtered and not reabsorbed or secreted. This is an example of ideal substance X. Inulin can be injected and its plasma clearance determined as a clinical means of finding out the glomerular filtration rate. Because all glomerular filtrate formed is cleared of inulin, the volume of plasma cleared of inulin per minute equals the volume of plasma filtered per minute, that is the glomerular filtration rate. Next situation. If substance is filtered and reabsorbed, but not secreted, its plasma clearance rate is always less than glomerular filtration rate. Some or all of a reabsorbable substance that has been filtered is returned to the plasma. Because less than the filtered volume of plasma will have been cleared of the substance, the plasma clearance rate of a reabsorbable substance is always less than the glomerular filtration rate. For example, the plasma clearance for glucose is normally 0. All the filtered glucose is reabsorbed with the rest of the returning filtrate, so none of the plasma is cleared of glucose. For a substance that is partially reabsorbed, such as urea, only part of the filtered plasma is cleared of the substance. With about 50% of the filtered urea being passively reabsorbed, only half of the filtered plasma, or 62.5 milliliters, is cleared of urea each minute. And third situation, if substance is filtered and secreted, but not reabsorbed, its plasma clearance rate is always greater than glomerular filtration rate. Tubular secretion allows the kidneys to clear certain materials from the plasma more efficiently. Only 20% of the plasma entering the kidneys is filtered. The remaining 80% passes unfiltered into the peritubular capillaries. The only means by which this unfiltered plasma can be cleared of any substance during the trip through the kidneys before being returned to the general circulation is by secretion. An example is hydrogen ions. Not only is filtered plasma cleared of non-reabsorbable hydrogen ions, but the plasma from which hydrogen ions is secreted is also cleared of hydrogen ions. For example, if the quantity of hydrogen ions secreted is equivalent to the quantity of hydrogen present in 25 milliliters of plasma, the clearance rate for hydrogen ions will be 150 milliliters per minute at the normal glomerular filtration rate of 125 milliliters per minute. Every minute, 125 milliliters of plasma will lose its hydrogen ions through filtration and failure of reabsorption, and an additional 25 milliliters of plasma will lose its hydrogen ions through secretion. The plasma clearance for a secreted substance is always greater than the glomerular filtration rate. Just as inulin can be used clinically to determine the glomerular filtration rate, plasma clearance of another foreign compound, the organic anion para-aminohippuric acid, can be used to measure renal plasma flow. Now let's talk about hormonal regulation. So previously we said that aldosterone stimulates sodium reabsorption in the distal and collecting tubules. So in the proximal tubule and the loop of Henle, a constant percentage of the filtered sodium is reabsorbed regardless of the sodium load. So the total amount of sodium in the body fluids, not the concentration of sodium in the body fluids. In the distal and collecting tubules, the reabsorption of a small percentage of the filtered sodium is subject to hormonal control. The extent of this controlled discretionary reabsorption is inversely related to the magnitude of the sodium load in the body. If there is too much sodium, little of this controlled sodium is reabsorbed. Instead, it is lost in the urine, thereby removing excess sodium from the body. If sodium is depleted, most or all of this controlled sodium is reabsorbed, conserving for the body sodium that otherwise would be lost in the urine. The sodium load in the body is reflected by the extracellular fluid volume. Sodium and its accompanying anion chloride account for more than 90% of the extracellular fluid's osmotic activity. Whenever we speak of sodium load, we technically mean salt load too, because chloride goes along with sodium. The sodium load is subject to regulation. Chloride passively follows along. Recall that osmotic pressure can be thought of loosely as a pulling force that attracts and holds water. When the sodium load is above normal and the extracellular fluid's osmotic activity is therefore increased, the extra sodium holds extra H2O, expanding the extracellular fluid volume. Conversely, when the sodium load is below normal, thereby decreasing extracellular fluid osmotic activity, less water than normal can be held in the extracellular fluid, so the extracellular fluid volume is reduced. Because plasma is part of the extracellular fluid, the most important result of a change in extracellular fluid volume is the matching change in blood pressure with expansion, increased blood pressure, or reduction, decreased blood pressure of the plasma volume. Those long-term control of arterial blood pressure ultimately depends on sodium regulating mechanism. Now let's talk about activation and function of the renin, angiotensin, aldosterone system. So this is the most important and best known hormonal system involved in regulating of sodium ions. This system is initiated by the juxtaglomerular apparatus, the specialized combination of tubular and vascular cells, where the tubule, after having bent back on itself, passes through the angle formed by the afferent and efferent arterioles as they join the glomerulus. The juxtaglomerular apparatus secretes the enzymatic hormone renin into the blood in response to a fall in sodium chloride, extracellular volume, and arterial blood pressure. These interrelated signals for increased renin secretion all indicate the need to expand the plasma volume to increase the arterial pressure to normal in the long term. Through a complex series of events involving the renin angiotensin aldosterone system, increased renin secretion brings about increased sodium reabsorption by the distal and collecting tubules. The ultimate benefit of this salt retention is osmotically induced water retention, which helps restore the plasma volume. Let us examine in further detail this mechanism that ultimately leads to increased sodium reabsorption. Once secreted into the blood, renin acts as an enzyme to activate angiotensinogen into the angiotensin 1. Angiotensinogen is a plasma protein synthesized by the liver and always present in the plasma in high concentration. On passing through the lungs via the pulmonary circulation, angiotensin 1 is converted into angiotensin 2 by angiotensin converting enzyme, ACE, which is abundant in the pulmonary capillaries. Angiotensin 2 is the main stimulus for secretion of the hormone aldosterone from the adrenal cortex. The adrenal cortex is an endocrine gland that produces several hormones, each secreted in response to different stimuli. Now let's talk briefly about atrial natriuretic peptide, which inhibits sodium reabsorption. Whereas renin-angiotensin-aldosterone system exerts the most powerful influence on the renal handling of sodium, this sodium retaining blood pressure raising system is opposed by a sodium losing blood pressure lowering system that involves the hormone atrial natriuretic peptide. The heart, in addition to its pump action, produces atrial natriuretic peptide, which is stored in specialized cardiac atrial muscle cells and is released when the heart muscle cells are mechanically stretched by an expansion of the circulating plasma volume when the extracellular fluid volume is increased. This expansion, which occurs as a result of sodium and water retention, increases arterial blood pressure. In turn, the main action of ANP is to inhibit sodium reabsorption in the distal parts of the nephron, thus increasing sodium excretion and thereby promoting accompanying osmotic water excretion in the urine. This natriuresis and accompanying diuresis indirectly lower blood pressure by reducing the sodium load and hence the fluid load in the body. In addition, ANP directly lowers blood pressure by decreasing the cardiac output and reducing peripheral vascular resistance by inhibiting sympathetic nervous activity to the heart and blood vessels. The relative contributions of ANP in maintaining salt and water balance and blood pressure regulation are presently being intensively investigated. So recent studies suggest that a deficiency of the counterbalancing natriuretic system may underlie some cases of long-term hypertension by leaving the powerful sodium-conserving system unopposed. The resulting salt retention, especially in association with high salt intake, could expand extracellular fluid volume and elevate blood pressure. And the last hormone to be discussed is vasopressin. So for water absorption to occur across a segment of the tubule, two criteria must be met. First, an osmotic gradient must exist across the tubule. And second, the tubular segment must be permeable to water. The distal and collecting tubules are impermeable to water, except in the presence of vasopressin, also known as antidiuretic hormone, which increases the permeability to water. Vasopressin is produced by several specific neuronal cell bodies in the hypothalamus and then stored in the posterior pituitary gland, which is attached to the hypothalamus by a thin stalk. The hypothalamus controls the release of vasopressin from the posterior pituitary into the blood. In a negative feedback fashion, vasopressin secretion is stimulated by a water deficit when the extracellular fluid is too concentrated, that is hypertonic, and water must be conserved for the body, and it is inhibited by water excess when the extracellular fluid is too dilute, that is hypotonic. And surplus water must be eliminated in urine. Vasopressin reaches the basolateral membrane of the tubular cells, lining the distal and collecting tubules through the circulatory system. Here it binds with receptors specific for it. So the binding activates the cyclic AMP, second messenger system, within these tubular cells. This binding ultimately increases permeability of the opposite luminal membrane to water by promoting insertion of vasopressin-regulated water channels in this membrane by means of exocytosis. Without these water channels, the luminal membrane is impermeable to water. Once water enters the tubular cells from the filtrate through these vasopressin-regulated luminal water channels, it passively leads the cells down the osmotic gradient across the cell's basolateral membrane to enter the interstitial fluid. The water channels in the basolateral membrane of the distal and collecting tubule are always present and open, so this membrane is always permeable to water. By permitting more water to permeate from the lumen into the tubular cells, the additional vasopressin-regulated luminal channels, those increase water reabsorption from the filtrate into the interstitial fluid. The tubular response to vasopressin is graded. The more vasopressin present, the more luminal water channels inserted, and the greater the permeability of the distal and collecting tubules to water. The increase in luminal membrane water channels is not permanent, however. So the channels are retrieved by endocytosis when vasopressin secretion decreases and cyclic AMP activity is similarly decreased. Accordingly, H2O permeability is reduced when vasopressin secretion decreases. These water channels are stored in the internalized vesicles, ready for reinsertion the next time vasopressin secretion increases. This shuttling of regulated water channels into and out of the luminal membrane under vasopressin command provides a means of rapidly controlling water permeability of the distal and collecting tubules, depending on the body's momentary needs. Vasopressin influences water permeability only in the distal and collecting tubules. It has no influence over the 80% of the filtered water that is obligatorily reabsorbed without control in the proximal tubule and loop of Henle. The ascending limb of Henle's loop is always impermeable to water, even in the presence of vasopressin. So this is all that I wanted to discuss for today's lecture. Thank you for your attention.